Hi guys, so back with biological molecules part 2. Here we go. Question number 11. A solution of starch is mixed with a solution of amylase. So what we need to understand here is amylase is an enzyme. It's an enzyme that is used to break down starch. Starch is a polymer of alpha glucose and it consists of two components, amylose and amylopectin. You might be confused with amylose and amylase. They are the different things. So amylose is a component of starch while amylase is an enzyme that is used to break down the starch. So our question is, which reagent should be used to confirm that a reaction has taken place, and what would be the appearance of the mixture when the reaction was complete? So the reaction here is amylase breaking down starch. So starch breaks down to give its residue, which is alpha glucose. Let's check out the options. Here we can cancel out option B and option C because beard solution that is used to test for proteins, ethanol is used to test for lipids, but starch is not a protein or lipid, it is a polysaccharide, so that's not done. So iodine and potassium iodide solution that is used to test for starch itself. So if iodine and potassium iodide solution gives a positive color and the appearance of the mixture is blue black, then it confirms that the reaction has not taken place because the starch has not broken down. So that is also not our option, and so our correct option is A, Benedict solution and brick red. So Benedict solution is used to test for reducing sugar, and alpha glucose is a reducing sugar. So the appearance of the mixture should be brick red if reducing sugars are present. Because starch is made up of alpha glucose molecules, when using Benedict solution as a reagent, the appearance of the mixture will be brick red because it gives a positive result. The most important thing here is first you need to cancel out all of the options that are obviously incorrect. So beard solution and ethanol, that was obviously incorrect because starch is not a protein nor a lipid or a fat. So that is not correct. And iodine in iodide solution, that you have to think a little bit, but then you will figure out because we need to confirm that a reaction has taken place. And amylase is an enzyme that is used to break down starch. So what would be the reaction? Breaking down a starch. We need to confirm that a reaction has taken place, and the reaction would not have taken place if starch was not broken down. So your answer will be A. So in this way, for any other kind of questions, you can first eliminate the options that are obviously incorrect and then use your thinking skills because you have minimal options left and you can easily make a correct guess if you have to, or you can easily find out what the answer is. Question number 12. The movement of water through the vascular tissue in plant relies on which property of water? We need to consider the properties of water that will allow the movement of water through the vascular tissues. Water, being a good solvent of ions and polar molecules, has nothing to do with its movement, so you can cross that out. And changes in density with temperature, that also does not help in the movement of water, so you can also cross that out. High specific heat capacity is the amount of heat that is required to increase the temperature of water by 1 degree Celsius. It has nothing to do with movement either, so you can cross that out. So you first eliminate all the options that are not feasible, and then you are left with D, strong cohesive forces between molecules. As discussed in the previous videos, cohesion is the attraction between like substances, and because water molecules are attracted to each other, they pull each other when they are moving upward through other factors as well. There are other factors like capillary action, which we will discuss in chapter 7. So for number 13, which statement is true for cellulose but not true for protein? So when you come across these types of questions, you have to consider both of the clauses, both of the conditions, right? So your option must be true for cellulose and it should not be true for protein. So first, let's look at what cellulose and proteins are. Okay, so cellulose are polysaccharides and they are made from beta glucose monomers, right? And proteins, they are polypeptides and they're made from amino acids. So let's look at the option from backwards. So it may be a structural component. It is true for cellulose. Cellulose is mostly a structural component. However, that is also true for protein. Protein can be a structural component. So it does not satisfy both of the conditions. So we eliminate option D. So it is used as an energy source. Cellulose is not used as an energy source because its function is more mechanical and it provides strength and mechanical support to plant cell walls. This statement is not true for cellulose. We eliminate that as well. So another option is it is synthesized from identical subunits. Cellulose is synthesized from beta-glucose monomers, and all beta-glucose monomers are identical. They do not vary from one monomer to another, right? But however, proteins, they are made from amino acids, and as we have discussed in the previous video, if you have not watched it, please do. Amino acids, they have the same basic structure, however, there is a difference. R group or the side chain. So that R group changes from one amino acid to another, and that means it creates different amino acids. So protein is not synthesized from identical subunits. This statement, so statement B, it is true for cellulose, however, it is not true for protein. So it could be our answer. Now let's look at the one last remaining option, and that is it is found in cell surface membranes. It's not true for cellulose because cellulose is not found in cell surface membranes. So you can cross it out because it already just satisfies a condition. Our answer is option B. Next, which level of protein structure maintains the globular shape of enzymes? And for these types of question, you have to understand the different levels of protein structure. So there are four different levels: primary, secondary, tertiary, and quaternary. The primary level of protein structure involves the number and sequence of amino acids in the protein. The secondary level of protein structure involves the alpha helix or beta pleated sheet. And the tertiary structure refers to the 3D shape, and globular shapes and fibrous shapes, they are the two types of 3D shapes that we'll have to study about. And the quaternary structure is only considered if there is more than one polypeptide involved in making the enzyme. And because enzymes are proteins, all of these apply to enzymes as well. And from this, we can see that the tertiary level of protein structure maintains the globular shapes of the enzyme. So our answer will be C. Question number 15. What is the function of the iron in a hemoglobin molecule? I'll tell you the basic structure of a hemoglobin molecule. So the basic structure of hemoglobin molecule is that it has four polypeptides, two alpha chains and two beta chains, and each polypeptide has a hem group, which is a non-proteinous structure, and the hem group is the iron. So what is the function of the iron in the hemoglobin molecule? So let's look at the options. It undergoes reversible oxidation and reduction. That is not the function of iron in the hemoglobin molecule. Oxidation and reduction, they are the process by which you lose or gain electrons, and the iron in the hemoglobin molecule is not related to the loss or gain of electrons. The main function of hemoglobin molecule is to carry oxygen. You can easily identify it as option A because it binds oxygen to the hemoglobin molecule. That's the function of the iron in the hemoglobin molecule. The function of hemoglobin is to carry oxygen, and that is facilitated by the iron that is present in the hemoglobin molecule. Now, if you do not know the function of the iron, you can go by the process of elimination. First, eliminate, it undergoes reversible oxidation and reduction. It stabilizes the quaternary structure of the protein. Any structure of the protein is stabilized by the bonds that are present in them, not by iron or any other kind of elements present in them. It's because of the bonds. So that's also not our answer. And it makes the hemoglobin more soluble in the red blood cell. That is also not our option. So our option is A. Now question 16. During the production of apple juice, enzymes are used to break down the components of the cell walls. Which carbohydrate will be produced by this hydrolysis? If you remember, when I was talking about cellulose before in the question about cellulose and proteins, I mentioned that the main function of cellulose is to provide mechanical strength and structural support to plant cell walls. And I also mentioned that cellulose is a polymer of beta-glucose. It is formed by joining many beta-glucose molecules together. So when you approach this question, you have to read through the keywords very importantly. So during the production of apple juice, enzymes, they break down the components of cell wall. So there are many different components of cell walls, but cellulose is present in cell walls of plants. So if enzymes are breaking down the components of cell walls, then cellulose will also be broken down. Which carbohydrate will be produced by this hydrolysis? So cellulose breaking down will release beta-glucose because cellulose is formed by joining many beta-glucose molecules. So our option here would be beta glucose. Our straightforward answer would be beta glucose. After the hydrolysis, cellulose will not be present. It will only be beta glucose. So that is also not an option. But however, here you can be confused between alpha glucose and beta glucose. In this case, you cannot, if you do not know that beta glucose is the answer, you cannot eliminate alpha glucose as well. And you might be confused. So you will also have to remember the key concepts. For this, you have to practice a lot. If you come across similar questions again, then you'll remember it. And if you practice it again and again, then you'll remember it if this question comes on your exam. Now let's deal with question number 17. The diagram shows a part of a macromolecule in the form of triple helices, lying side by side by covalent with covalent cross links between them. So here, underline the keywords in the first place. When you're reading a question, keep a pencil and underline the keywords. So here, one of the keyword is triple helix. Another is it's a macromolecule. They're lying side by side and they have covalent cross links between them. So these four are your keywords which will help you identify what molecule this is. So for this, you will also have to study the features or characteristics of all the macromolecules. And if you remember properly, then triple helixes are present in collagen molecules. And if you do not have an idea of what the answer could be just by reading the question, you can take help of the diagram given here. There are similar diagrams in your textbook. So by remembering the diagrams from your textbook, like you can see that this is similar to collagen. So our answer is collagen. It's not cellulose. Cellulose does not form any triple helixes, and it's not glycogen because glycogen is a branched molecule, and this is not branched. It's also not triglyceride because they do not form triple helixes and they do not lie side by side like this. So collagen is your answer, and for this, also, again, you have to and for this, again, you have to remember the characteristics of all the macromolecules that are present. In biology, it's a very content-heavy subject. You can use your skills, but knowledge is also equally important. You can use your skills like eliminating or making an educated guess, but then again, knowledge is very important for all of those as well. You need to understand the concept of the topics that you're studying about. Okay, now question number 18. Which of the following is a polysaccharide that is present in human muscle? But you can easily cross out amylose because amylose is a component of starch and starch is only present in plants and not in animals. Humans are animals, so starch is not present in human muscles, so amylose will also not be present in human muscles. Collagen is a protein that is mostly found in skins, so it's not present in human muscle. Hemoglobin is present in red blood cells because its function is to carry oxygen. So that is also not our option, and we are left with glycogen, and that's our correct answer because glycogen is an energy store. It's similar to starch, but it's only present in animals. So that's our correct answer. Moving on to question number 19, which combination describes a triglyceride? So there are two options, hydrophilic and soluble in alcohol. So for this, you have to remember the characteristics of a triglyceride. So don't get confused between phospholipids and triglycerides. The triglycerides are hydrophobic and are insoluble in water. Phospholipids that are hydrophobic, they have both hydrophobic and hydrophilic parts, and they are soluble in water. So it's hydrophobic. That means that it is not attracted towards water, and something that is not attracted towards water and cannot form hydrogen bonds with water cannot be soluble in water. So, looking at these characteristics, you can make a guess. Let's first eliminate the answers that are not possible at all. So A is not possible and C is not possible because triglycerides are not hydrophilic. The remaining options are B and D. So triglyceride is insoluble in water, but it is soluble in organic solvents. And alcohol is an organic solvent, so triglyceride will be soluble in alcohol, so you can cross out option B. Triglyceride is not hydrophilic, but is soluble in water, so our correct option will be D. Okay, question number 20 with so many diagrams. So the diagram shows 4 different molecules and which shows the correct information about the molecule or molecules. Okay, so let's look at the options first. We'll come back to the diagram. Contains a carboxyl group, forms 1,6 glycosidic bonds in glycogen, forms peptide bonds by condensation. It would be a great advantage if you can identify the molecules from their molecular formulas. That would be great. Like, for example, this is alpha glucose, this is beta glucose, it's an amino acid. This one is also an amino acid. It will be great if you can identify molecules like this through their molecular diagram. And it's very easy to do that as well. It's not very difficult. Like for these amino acids, you just have to remember the basic structure with NH2, NH2, COOH, COOH, hydrogen, hydrogen, and the alpha carbon in the middle, and alpha carbon over here. Even if you do not know the specific names of amino acid, it does not matter if you can identify, oh, this is an amino acid by looking at the molecular structure. Because it's not quite difficult because they are identifying features like the NH2, NH2, COOH, COOH, H, and the central carbon. It's common in everything, and the only thing that is different from one amino acid to another is this part, which is known as the side chain or the R group. And for alpha glucose and beta glucose, look at this OH over here. This OH, if it is below the CH plane, this is the plane, this is the CH plane. If it is below the CH plane, then it is alpha glucose, and if it is above the CH plane, it's above the CH plane, right? In this one, in R, so it is beta glucose. So you can identify your molecules like this. It requires consistent practice and drawing out the molecules. It can help you identify it. So practice it. If you cannot identify it just from the looks of it, you can practice it, and once you get the hang of it, then it becomes easy. So let's look at the options. Before looking at the options, let's look at the descriptive content that is given here. So contains a carboxyl group. Whenever there is any carboxyl group, COOH is a carboxyl group. It's Q and S because they have the COOH group. COOH. They're also COOH. P and R do not have COOH. So for this section, our answer can either be Q or S. So forms 1,6 glycosidic bonds in glycogen. The things that you have to study about starch, glycogen, and cellulose, among them, starch and glycogen. Both of them are polymers of alpha glucose, and only cellulose is a polymer of beta glucose. So if glycogen is mentioned, and forms 1,6 glycosidic bonds in glycogen, it will be alpha glucose. And as I've mentioned earlier, P is the alpha glucose. So our option for this is B. And another is forms peptide bonds by condensation. And peptide bonds are present in proteins, and proteins are made from amino acids. So our possible answers for this is Q or X. Because we know what are possible answers for each. So if it is not the possible answer, you can cross them out. For example, in this contains a carboxyl group, P and R are not a possible answer. So you can cross out A and C. Forms 1,6 glycosidic bond. P is a possible answer, but R isn't. So B also gets crossed out. So now we're left with answer D, which is our correct answer. And we can also look at here. Oh, it says and. So Q and S form peptide bonds, which we figured out earlier before looking at the option. And P is the correct option for this column, and for this column, Q or S is the correct option. So here S is present, so it is the correct option. So like this, our correct answer for 20 is D. So that's it for this video. We have more topicals to solve, so look forward to other videos. And if you enjoy it, like and follow or subscribe or do whatever you want. And I hope it helps you. Thank you for watching.